So Tricia received her PhD at Yale. After postdoctoral work, she moved to Brown University, right? Where she was a professor. Then she moved to the University of Arizona to become the head in the vernacular of the university of the Department of Molecular and Cellular Biology. Tricia is currently the Dean of the College of Natural Sciences and the Associate Chancellor for Strategic Academic Planning at the University of Massachusetts Amherst. Now, I can go on and tell you about Tricia's many things. I'm just going to tell you about one story about all her awards and publications and all that. I'm not going to do that. You can look it up if you want. I'm going to tell you one story though, and nobody knows the story. So I was the acting head of the university, of the Department of Molecular and Cell Biology. And I was the head of the search committee that recruited and hired Tricia. So when we offered her the contract, she calls me. And she says, Johnny, if you want to be chair, continue from acting chair to be permanent chair, I will withdraw my application. That's what she said to me. I said, Tricia, I have no interest in being the head of the department. She said, then I want you as associate head. I said, Tricia, I don't want to be associate head. She said, no, I need you because you know the culture, you know the people. The next 5 years, and I say it very objectively, was the best 5 years for the department. The growth was unbelievable. The change, the organization, the vision, it was amazing. And a lot of what I do now, and this is honestly, I'm not being, I learned from you, Tricia. Okay, the good stuff. So anything good, I learned from her. Anything bad is just all me. So without further ado, Tricia, thank you so much for accepting, and I'm looking forward to your presentation.
Thank you so much, Johnny, and thanks so much for joining me today. I'm gonna share my slides. And, okay, everybody see that okay? Thanks so much for the invitation to talk to you today about prions and their really fascinating biology. Prions were first described to explain a group of diseases known as transmissible spongiform encephalopathies, which are a group of infectious diseases. And the most well-known versions of these are mad cow disease in cattle, scrapie in sheep, and chronic wasting disease in deer and elk. And these diseases all share a common pathology, and that is the appearance of vacuoles in the brain of animals who are afflicted with these disorders in comparison with healthy brain tissue. Now, scrapie was first described in the literature in the 18th century, so these diseases have been around for a really long time. But it wasn't until the late 1950s when Hadlow, who was a veterinarian, realized that that same spongiform pathology that I just showed you was also characteristic of a number of human diseases, including Creutzfeldt-Jakob disease, Kuru, and fatal familial insomnia. Now, this realization that there were transmissible spongiform encephalopathies, or TSCs, as I'll call them, in humans, opened up a whole new understanding of these diseases, because Kuru, like the animal diseases that I described, was an infectious disease, but the other human diseases appeared in different forms. For example, some were inherited and some were sporadic. That is, they just appeared and we don't know why they appeared. Now, this particular collection of characteristics is unique if you think about the ways we understand diseases in general. So there are infectious diseases such as SARS-CoV-2, which we all know well, given our current time and history, and Salmonella, which are characterized by the infection of an individual with a microorganism, either a virus or a bacterium. And this is associated with the transmission of a piece of foreign DNA or RNA, which is the genome of the microorganism, now enters the body and allows this organism to replicate and cause disease. The other type of diseases that we're really familiar with, of course, are heritable diseases such as sickle cell anemia or Duchenne's muscular dystrophy. And these are associated not with some kind of foreign gene, but rather through a mutation in a gene in our own DNA, in our own chromosomes, which replicate that trait and allow us to transmit it to our progeny. So if you think about diseases in this frame as being either infectious or heritable, it's hard to imagine a scenario where they could be both at the same time. Like, how could you transmit sickle cell anemia to another person? It is for this reason that the TSCs have these unusual characteristics in humans that was first proposed that they represent a new type of pathogen and one that's really serious. So the TSCs have very strong pathology. They're rapidly progressive. So decline happens very quickly when someone acquires one of these diseases. And up until this point, they have always been fatal. And even beyond that, they are scary if you actually think about it. They are very resistant to inactivation. So there have been studies showing that, for example, scrapie persists in a barn for more than a decade and can be infectious to a new animal that's introduced, and that these diseases can be transmitted from one individual to another on surgical instruments that had been sterilized in an autoclave. So this pathogen, whatever it is incredibly resistant to inactivation. So given this unusual collection of traits, it's probably not surprising to you that the Nobel Prize has been awarded for prions actually twice. The first time was in 1976 to Kartin Gajicek, who showed that biopsy material from a patient with Creutzfeldt-Jakob disease could cause that same spongiform encephalopathy when injected into a chimpanzee. So he effectively showed that a genetic disease could be transmissible. The prize was also awarded again in 1997 to Stanley Prusiner, who showed that these diseases were caused by a new type of pathogen, which he called a prion, for proteinaceous infectious particle. And a prion is essentially an infective protein, a protein that is infectious. We now know that the prion protein in mammals, this includes animals and humans, which we call PRP for prion protein, is a complicated molecule. It has a number of repeats. It has a portion of the protein that is really resistant to degradation, which explains the persistence of the pathogen in nature, in the environment, and it's membrane associated through the GPI anchor. Most importantly, the human diseases that have fallen under the umbrella of TSC have all been shown to have mutations in the gene that encodes for the prion protein, which we call the PRNP gene. So that's pretty strong evidence that the protein is associated with these diseases. But as people continue to study these disorders, another complication arose. So we would predict if this protein caused disease that it would be present in individuals that have that disease. And that's certainly the case. The conundrum arose when we looked for this protein in healthy individuals and realized that the protein was also expressed in those individuals. So that didn't make a lot of sense. How could a protein cause disease but be present in both healthy individuals and those with disease? Well, it turns out that there's yet another wrinkle in this story, and that is, unlike most proteins, the prion protein can fold into at least two different shapes or conformations. One is associated with the healthy state, and the other is associated with the disease state. And so you can think about TSEs as a protein misfolding disease. That is, the protein is present all the time, but it is the shape that determines whether a person is afflicted with a disease or is healthy. Now, this may seem strange for various reasons, so I want to just pause and share an analogy with you. and I'm a knitter, so this is why I'm showing this analogy. So I can take a skein of yarn and I can knit that skein of yarn into a pair of gloves, or I can take that same skein of yarn and then knit it into a hat. Each of these, the gloves and the hat, are created from the same yarn, but because I folded them in a different way, they have different functions. And in the same way, the prion diseases can be explained. So if the protein folds into one shape, it will have one function. And if it folds into a different shape, it has a different function. And in those cases, healthy state or a disease state. Now, that provides a framework for us to think about this. But if you think about it in more detail, there has to be something else going on. And in particular, if this protein can fold in either shape, just randomly as it's being made, we'd never create distinct outcomes, right? So if one molecule folds this way and another folds this way, what you'd wind up with is a mixture of different forms in any individual. Instead of two distinct states, you'd be somewhere in between here. So there's got to be another piece to this story that explains why you can get a disease state or a separate healthy state. And the answer to that is that there is a mechanism to ensure that once the disease-associated fold or conformation arises, that the newly made protein preferentially folds in that same way. And so once this appears, it can essentially self-replicate by folding all the newly made protein in this direction. And that process hadn't been described before. So how does it work? Well, central to this conformational self-replication is the fact that the prion protein can aggregate. And what I mean is that the individual molecules of the protein come together and they form these long linear fibers. And we can see these if the protein is just purified and studied in vitro in a test tube, or we can isolate samples from an individual with disease, and you can see these fibers present as well. And these structures are called amyloid because they have a very particular organization to them. Now, the assembly of these amyloid fibers or aggregates is an inherent property of these proteins. So I can take a test tube and put purified protein in it in the non-prion or non-amyloid state. And it will spontaneously refold to the other conformation, which I'll call the prion conformation. And when this happens, this new fold has a really high propensity to self-associate into an aggregate. And you can think of this little diagram, which I'll show throughout this talk, as a cross-section of one of these fibers. So at the end, for example. And this surface has a really unusual property in that it can act as a template, and it can take protein in the non-prion state, bind to it, change its shape, and incorporate that protein to make the aggregate bigger. So that's how they grow in this long linear structure, because you keep adding new protein onto the end. Okay. So this process explains how a conformation can replicate and how these diseases can become progressive. But why does this happen? We know that normally inside cells, proteins misfold all the time, and the cell is able to deal with them through quality control pathways. And we generally think of protein quality control in two ways. One is that there are enzymes known as proteases, which degrade proteins that are folded incorrectly. And the other is chaperone proteins, which are enzymes that take misfolded proteins and refold them. And so if these systems exist in cells, why is it that amyloids arise and persist and cause these diseases that can't be cured? When I first started working in the field, the general idea was that the quality control pathways that were present weren't able to act on amyloids because these complexes are so stable that cellular enzymes just couldn't touch them. And this is the main reason that we thought that prion diseases are incurable. But I'm a little stubborn, and I want to go back to my knitting analogy. I know that if I have knitted this hat and I don't want it anymore, I can unravel it and take the yarn back and make a pair of gloves out of it. So why can't we do that with these proteins? Why can't we unravel them and refold them into a new shape and cure these diseases? Well, up until this point, I've been talking to you about the TSCs and the prion protein that's misfolded to cause them. But there's actually now a whole set of diseases that are known as protein misfolding diseases. There's more than 50 in humans alone. Some of these you have likely heard of, including Alzheimer's disease, Huntington's disease, and Parkinson's disease. And these are all diseases that are associated with amyloid, but in each case, a different protein is misfolding. But this amyloid conformation is not only associated with diseases. So we know now that there are a number of different phenotypes in various organisms that are associated with the amyloid state. And they can do things like store melanin in your skin or hormones. They can form biofilms in bacteria. They can regulate transcription and translation in microorganisms. And these type of amyloids are called beneficial amyloids. And the really important thing to know about them is they are all forming some kind of a regulated function. And that raises the idea that they can be reversed. And so for the rest of my time with you today, I'm going to talk about a particular prion, which is called sub-35, that's present in baker's yeast. And what we've learned by studying that about whether amyloids can be reversed or not. So sub-35 is a protein that has this general structure. It has a domain at its end terminus, which we call the prion domain. And this part of the molecule can change its shape. And this is fused to an enzyme, which is responsible for hydrolyzing GTP in the cell. And this protein functions normally in translation termination, and it specifically stimulates release of the polypeptide when translation is finished. Now, sub-35, like all good prion proteins, can exist in two forms. Its normal state, which we call Psi minus, and its amyloid state, which we call Psi plus. And it makes the same type of fibers that I showed you before for PRP, these long linear amyloids. And these amyloids, just like was true for PRP, are assembled in this pathway of polymerization that's nucleated by an existing template. So we call this nucleated polymerization. In addition to studying this protein in vitro, we can also study it inside of yeast cells. And if we tag it with a fluorescent protein, you can see that in Psi minus cells where the protein is soluble and normal, you get this kind of diffuse pattern throughout the cell. And in Psi plus cells where the protein is aggregated, you see these small foci all throughout the cell. Now, we can also study the process of nucleated polymerization inside cells. So we know if we introduce an aggregate of sub-35 into cells, it will take the normal form of sub-35 and pull it into the aggregate, changing its shape. And we can keep this cycle going in a growing yeast cell if we keep making sub-35 protein so that there's always a source for conversion. And if there's a process to fragment the aggregates as they grow into smaller pieces. And what this does is create more templates so that you can keep adding on the protein as it's been made. And it also keeps them small enough that they can be transmitted to daughter cells during division. So both the mother cell and the daughter cell have an aggregate, and that persists in the population. Now, this is a lot of complicated dynamics that are going on inside the cell at the protein level. And we can essentially have a proxy for what's happening here by looking at the phenotype of yeast. So in the lab, we use yeast strains that have a mutation in the AD1 gene that introduces a stop codon in that gene. And you may remember that the sub-35 protein works at stop codons. And so when it's present in its psi- state, it's fully functional, and this premature stop codon is recognized. And so the full length protein isn't made. And that's seen as a mutation. And what happens is the yeast form red colonies on a plate. And when sub-35 is aggregated, its function is reduced because it's in this aggregate. And the ribosome can read through the stop codon and make the full-length protein. And that creates more of a wild type state. And the yeast colonies appear white on the plate. Okay, so we can read out what's happening at the protein level by looking at the colony color. But it's important to remember that even though these colonies look really different, these strains are genetically identical to one another. The only difference is what shape the sub-35 protein is in them. And the really interesting thing about this is, unlike the diseases that I described in animals and in man, these phenotypes can switch back and forth at a really low frequency. About 1 in 10 to the 8 cells will change its phenotype, but it can happen in either direction. Even though this happens really infrequently, it's exciting because it raises the possibility that the protein may be able to be refolded. And so we really wanted to understand how these transitions happen. Now, 10 to the 8th is really infrequent. And so we use all kinds of tricks to try to understand how this happens by making it happen more frequently. And that includes mutations, changes in conformation or the environment or gene expression. We can use inhibitors, and we can introduce other amyloids into the system. And this allows us to study these processes. So what we really wanted to know is, if we have a cell in which the amyloid form of sub-35 is replicating, can we cure it? That is, can we take the protein that was previously aggregated and refold it into the normal state? So a lot of what I'll tell you about today is our studies of this process and what we learned from it. And we also have studied the reverse process, where you take a cell that doesn't have an amyloid and watch and understand what happens when that amyloid appears. So I'll tell you a little bit about that as well. Now, before I get into the details, I want to tell you right up front what we learned, and it's very exciting to me. First, we found that chaperones, which were those enzymes that refold proteins, can actually disassemble amyloid. So we can take it from the amyloid state and return it to the normal state. Another thing we learned is that this process of disassembly is really dependent on what the conformation of the prion protein is in the amyloid. So some are sensitive and others are not. And by studying that, we learn more about how these diseases arise and be cured. And the third thing we learned is that chaperones also regulate the appearance of the amyloid state. And we can think about how to block the appearance of these diseases based on this knowledge. So I'll go through each of these in turn in our remaining time, but I'm going to start with the first, which is the fact that chaperones can disassemble amyloid. Now, I mentioned before that there are quality control pathways that deal with misfolded proteins. What I didn't tell you is that this process is adaptive. That is, if we do something to increase protein misfolding, for example, like heat shocking cells, the cell responds by changing what proteins are made. So this is the normal complement in flies or in yeast. And then when we heat shock, different proteins are made. And these are called heat shock proteins. And what they do is increase chaperones and proteases so that the misfolded proteins get resolved and the cell returns to a normal state. So we wondered, what happens if there's an amyloid if we ramp up these protein quality control pathways with a heat shock? So the pictures of yeast that I've shown you are grown at 30 degrees, which is the normal temperature for growing yeast cells. And so what we did was shift the temperature to 40 degrees for just a half an hour. And what we saw was something amazing. that there's a high frequency of switching from the Psi plus to the Psi minus state. That is, these cells were cured by this heat shock, and we really wanted to understand how that happened. Now, this cycle that I described to you before has many steps to it, and you can imagine that if you interfere with any of these steps, you're not going to be able to sustain the prion state. But what we were really interested in was asking if you could disassemble amyloid. And one thing that I didn't tell you was that one of these heat shock proteins called HSP104 functions to do this fragmentation step. That is, it breaks big aggregates into smaller ones. You can see that if we run the aggregates on an electrophoresis gel. So normally they have a smear of about this size, and if we inhibit HSP104, they get bigger. This fragmentation step is so important that if we get rid of HSP104, the strain turns from Psi plus to Psi minus, that is, it's cured, and it can never reacquire the prion state. So what this tells us is that chaperones can act on amyloids, but maybe they're just not very good at it. And so what we wanted to know is, if we took a cell and heat shocked it, is the activity of HSP104 ramped up so much that it disassembles the amyloid? And that seems to be the case. So if we look at how much soluble sub-35 is present in cells, you see as you increase the temperature at which those cells are grown, the amyloid disassembles and get more and more soluble sub-35. Now, if we use this highest temperature at 40 degrees as a starting point, if we inhibit HSP104 during the heat shock, that resolubilization that we saw here is blocked, and the protein stays in the aggregate. So this suggests to us that even though HSP104 can't normally disassemble the amyloid aggregates, if we increase its activity, it can take it all the way down to the soluble state. This was really exciting, and we thought, well, this is just way too simple. Why doesn't the cell just make more HSP104 so this never happens to begin with? And so we wanted to understand on a cellular level where this was happening. And we used a technique called flow cytometry that allows us to look at a large population of cells and how the protein is distributed in that population. So if we look at the normal growth temperature at 30 degrees, and here we've tagged HSP104 with the fluorescent molecule, you see that all the cells in the population fall in a narrow distribution of HSP104 levels. But when we heat shock, we get more HSP104, so this curve shifts to the right, but it also gets very uneven. And you can see that there are different populations of cells here that have different amounts of HSP104. When we isolate these different populations, what we find is the ones that have the most HSP104 have the highest amount of curing, and the ones that have the least HSP104 have the lowest amounts of curing. So why do cells have different amounts of HSP104, since this is so important for curing? We looked at this by microscopy, and what we found is shown here. So in a series of pictures that I'm going to show you, there are two cells. One is outlined in white, and it has started its division cycle already. And the other is outlined in orange, and it is not yet dividing. And here we are before we've done a heat shock. So after heat shock, what we see is that HSP104 levels increase. You see there's not much fluorescence here, and then it becomes bright. And then it localizes into these spots inside cells, which we know are the places where misfolded protein accumulates. So HSP104 is going to the misfolded protein. And what we find is, as time goes on and the cells divide, the one that started its division before heat shock, down here in white, most of the HSP104 stays in the original or mother cell, and there's not much in the daughter cell at all, because these aggregates are still present in this misfolded protein. And so already we have a difference in the population of cells where some have a lot of HSP104 and some have not very much. This orange cell then continues to divide, and you see that before it divides, it's gotten rid of most of the misfolded proteins. These dots are much less intense than they were here. And so by the time the cell divides, the HSP104 has gone into each compartment, and there's no one particular cell that has more than another. We can create this kind of state artificially using inhibitors. So alpha factor, for example, inhibits the cell cycle before it starts, and nicotazole inhibits the cell cycle late in the game, right before cell division. And so if we arrest cells with either of these compounds and then heat shock them, we can see if these cells, which accumulate different amounts of HSP104, are differentially cured. And that is exactly what happens. So the cells that are not budded yet and can distribute their HSP104 have really low amounts of curing. And those that are late in the cell cycle and retain HSP104 asymmetrically account for most of the curing that happens in the population. So what this tells us together is that chaperones can disassemble amyloid, but it's really important how much of them are around and how they are localized. And so cell division is really acting against the protein quality control system here by distributing the chaperones among cells in the population. Now that we know that amyloids can be disassembled, what determines how efficiently that happens? And one of the things we found is conformation. I mentioned it to you before, we've been really talking about amyloid existing in one conformation, but these proteins can adopt multiple different conformations, all of which are capable of forming amyloid. And these different conformations have different biological effects. In the case of PRP, they lead to the accumulation of protein aggregates in different parts of the brain, and that leads to different symptoms for the diseases. In the case of yeast, these different conformations inactivate the protein to different degrees in translation and termination, and we can follow that at the colony level by different shades of pink. Okay. Now, because these proteins, the protein in these different conformations is assembled into amyloid, we also know that it has an impact on how they are processed by chaperones. So for example, in the strong side case, we know that its conversion is slow, but its ability to be fragmented is faster. And the opposite is true for the weak side state, where conversion is very efficient, but fragmentation is very slow. Now, based on what I told you about heat shock, where if we have high levels of HSP104, we can completely disassemble the amyloid, we might predict that amyloids of different conformation that are fragmented at different rates might be cured at different rates. And that's exactly what we see. So up to this point, I've been talking to you about weak sign. That's the one that's really efficiently cured by heat shock. But if we look at strong psi under the same conditions, there's essentially no curing at all. But the problem is this doesn't make sense because strong psi is the one that's fragmented well, and weak psi is the one that's not fragmented very well. And we would expect the opposite here. So these observations suggest that just the rate at which chaperones act on these amyloids can't explain the curing. But is this actually true or not? This cycle that I've been talking to you about has a number of different steps that happen at distinct rates. So for example, synthesis of the protein, conversion to the amyloid state, fragmentation, and transmission. And because these all happen at rates and are connected to each other, we can make a mathematical model of them. And when we make this kind of a model, what you get here, what I'm plotting is fragmentation rate versus conversion rate. You get this kind of curve, which tells us under what combinations of these rates can the prion exist. So in this case, anything inside this curve is compatible with the strong state. And we can do the same thing for the weak state, which gives you a different curve here shown in red. The problem is, according to this model, weak psi should be stable at higher rates of fragmentation than strong psi. And that's the opposite of what we see. Weak psi is more sensitive to fragmentation than strong psi. So this suggests that there's something that we don't know about the system, and we wanted to try to figure out what that was. So if we think about how this happens, you have an aggregate that can progressively add more protein to it and grow in size, this nucleated polymerization process. But a key component of this that we haven't talked about is that there's some minimum size to the aggregate that is stable, like a certain number of molecules have to come together to form an aggregate. So if we just say that that minimum number is 4, something really important happens when that aggregate doubles in size to 8. And that is, this aggregate, if it's fragmented once, will produce aggregates that are bigger than 4, and so they can survive. If at any step before it doubles, that aggregate is fragmented, the protein will return to the soluble state because you will create aggregates that are smaller than 4 and are therefore not stable. So you can see this kind of graphically in this prediction. So the bigger the minimum aggregate size, the larger it has to grow before it can reach a state where it would survive a single fragmentation event. So based on this concept, we wondered, well, how does minimum aggregate size affect curing? So in the model that I showed you before, we had weak psi and strong psi have the same size aggregates. And so we wondered, well, what would happen if we said weak psi has a bigger minimal aggregate size? And something really remarkable happened. The curves shifted relative to each other. So now strong psi is stable at higher fragmentation rates than weak psi. And that could explain this curing that we see. Okay, that's a nice prediction, but we have to prove that it's true, or at least test if it's true. And we can do that in yeast cells if we stop synthesis of the protein. All of the aggregates will just keep getting, that are already there will keep getting fragmented. And first they'll get smaller, and then eventually they'll get completely disassembled. And you can see this in the mathematical model. In the light color here is a distribution of aggregate sizes where we start. And then over time, you see there are fewer and fewer aggregates because they're being disassembled. But something really important happens. They never get smaller than the smallest size that is stable, because once they hit that threshold, they completely fall apart. So you don't see anything smaller there. So this is a prediction from the model. And that's actually what we see in yeast cells. So if we stop new protein synthesis, the aggregates get smaller over time. And that's true for strong or weak psi. But the lowest size that they attain is different. So strong psi can be smaller than weak psi. And that's exactly what our model predicted, and helps us to explain why weak psi is cured at higher fragmentation rates, but not strong psi. So this suggests that the conformation is really important. And the reason it's important is because it defines the minimum aggregate size that can be stable. Okay. So that are two things that we learned about curing. Now I want to spend just the rest of the time talking about how chaperones can affect the rate of appearance of new psi. So again, this is really just the opposite process of curing, in which we look at the emergence of the amyloid state. So normally, prions can arise spontaneously, which I talked to you about, but it's really infrequent. And we can make that happen more frequently if there's already another amyloid present in the cell. And the one that we typically use is called PIN for psi inducibility. And it's determined by the RINC1 protein, which forms the amyloid. You can see how dramatic this effect is here in this experiment, where we plate yeast cells and select for growth of the psi phenotype. If PIN is not there, it hardly ever happens. And if PIN is there, it happens very frequently. So we wanted to understand, how does PIN promote Psi appearance? Now, this model that I showed you before, that you could disassemble aggregates if you had enough heat shock protein, we wondered if that could affect appearance of Psi. So under normal conditions, the protein will just spontaneously misfold, but it's not very abundant at that point because they're just appearing. So we wondered if maybe the normal levels of HSP104 were high enough to return those newly appearing aggregates to the Psi minus state. The question is then, what is PIN doing in this idea? Well, it turns out PIN is, because it's an amyloid, is also acted on by HSP104, which fragments it. And the model here would predict that because PIN influences Psi formation, maybe HSP104 has a higher affinity for PIN. So if PIN is there, it can titrate away the HSP104 from its action on sub-35. And then these aggregates that disappear are stable and persist. So we wanted to test this idea. So we started first not by looking at appearance, but rather by looking at a cell that had both the sub-35 amyloids, or Psi plus, and the rank 1 amyloid, or PIN plus. We know that both of these types of aggregates are fragmented by HSP104. And we wanted to look at the competition between them by monitoring how much of the sub-35 aggregates persisted. So to start you off, this is a normal state. This is how the aggregates migrate on a gel. And we thought, okay, if these are competing with each other, if we limit the amount of HSP104, that might make them compete more strongly with one another. And based on our model, we would predict that PIN would win. So we expected sub-35 aggregates to become less abundant. And that's exactly what we see. When we lower HSP104 levels, the sub-35 aggregates disappear. Now, if this is due to competition, if we got rid of the PIN aggregates, the sub-35 aggregates should come back. And so if we get rid of the rank 1 protein, that's exactly what happens. The sub-35 aggregates reappear. Now, if they really need and are very sensitive to HSP104 levels, we should even make this more abundant if we return HSP104 to its normal levels. And that's in fact what we see. We see more aggregates under these conditions where there's no PIN and normal levels of HSP104. Okay, so that suggests that there is competition here. Now the question is that competition relevant to appearance of a new Psi aggregate? Okay, so again, the idea here would be that in the presence of PIN, the activity of HSP104 on these aggregates decreases and allows them to survive. Now, we can test this idea by making sub-35 fibers in vitro and then transforming them into yeast cells and asking, do these yeast cells become Psi plus? And so if we use different conditions, we can ask, do HSP104 and PIN affect the survival of the aggregates we put in, just in the way that I tested it in the previous slide. And here, what we're going to do is change the amount of aggregates or those fibers that we put into yeast cells and see how sensitive it is. The experiment looks something like this. So we can add different amounts of fibers to yeast cells and then monitor the percentage of them that acquire Psi. You can see in the white circles here, this is just a PIN minus state. And as you put more fibers in, you get, oops, apologies. Okay, as you put more fibers in, you get more Psi plus. Now, if PIN is present, shown in the black dots, you can do the same thing, but with less fibers. Here, the curve is to the left of the PIN minus state. We see the same thing if we lower HSP104 levels, in this case in the open squares. You can get more psi at lower amounts of fibrous transformed. So that suggests that both PIN and HSP104 affect the survival of the aggregates that we put into these cells. Now, if that's the case, if HSP104 is normally disassembling sub-35 aggregates and PIN is titrating it away, then this whole process should be really sensitive to the levels of HSP104. We can test that directly in cells that are acquiring psi for the first time. And we can make this happen more frequently by overexpressing the sub-35 protein. What we find is that you get a certain level of psi appearance by doing this. Now, if we lower the amount of HSP104, we should get more aggregates because they're not being disassembled and more psi. And that's in fact what we see. And then if we do the opposite, that is, we put more HSP104 into the system, it should cause these aggregates to be disassembled. And that is exactly what we see as well, that there's an inverse relationship between the levels of HSP104 and the amount of psi that appears. So together, this tells us that the appearance of the prion state is really sensitive to amyloid, to chaperones, and that that's affected by the presence of other amyloids which can titrate them away. So I started with this very simple idea that we should be able to take one state, unfold it, and refold it to cure. And it turns out that that is indeed the case, at least in yeast, but it's incredibly complicated. And it can only occur under certain conditions with many different factors influencing the outcome of that situation. Now, when I started working in this field, we thought about amyloids as different energetic states, with the amyloid fiber being the most stable. And the idea was that if you came from a normal state and actually converted into an amyloid, you'd be stuck here and never be able to exit that form. We now know that amyloids are very dynamic in vivo, and they're constantly being converting their conformation and assembling into aggregates, and those aggregates are being processed by chaperones continually. And what our work really says is that there's this really fine balance between the forward pathway and the backward pathway here. And normally the amyloid wins. But as we've gotten more sophisticated in our models, what we find is that there's so many different factors contributing here, where the prion state is viable in the red or where it's lost in the blue. And all of the states that are really biologically relevant tend to fall on this boundary, which suggests that the prion state is just on this edge of being able to survive, which really gives me hope that we'll be able to tip it off that edge. There are a number of factors that contribute here, some of which we talked about: prion conformation, the sequence of the protein, chaperone abundance and localization, other misfolded proteins, and what stage the cell cycle is at. So you might have realized by now that I really like to work on puzzles, and that's one of the reasons that I really love being a scientist. The other is that I get to work on these puzzles with an amazing group of people who are just as fascinated by these problems as I am. So some of the work I talked to you today about is the work of Courtney Clates, a former graduate student in my lab, Janice Vallely, a former postdoc in the lab, and Teal Brechtel, a former graduate student, and my longtime collaborator, Suzanne Cindy, who's a mathematician. And I also really want to thank all the agencies who have funded our work over the years. And you can learn more about our work and about me at our website. Follow me on Twitter, or send me an email. I'd love to talk to you about science or anything else. Thank you so much. I really appreciate your time.